Okay, and here we go. We're going to look at a pretty important topic for people who work in the medical field, and that's dealing with rigor mortis. What happens with rigor mortis? What happens following death to muscles? And it's if you've not seen it before, it can be a little scary to understand and to explain to people. But we'll look at what happens during a contraction cycle and rigor mortis in this video. So, how does myosin pull on actin to cause a contraction? Why is ATP needed? I think we'll see that here. And what is rigor mortis? What is going on and why does it occur? So let's start by looking at the contraction cycle. Oops, let me get rid of this. Um, let's start by looking at the contraction cycle. You need to understand what goes on during a contraction so you understand what goes on during rigor mortis. So let's start with this. What we have here is myosin, and notice this term here. It's in the high energy configuration. That means that myosin is ready to go. We've spent the energy. We put myosin in this really strong energy position, and in this case, it's already attached to actin. Remember that myosin always wants to attach to actin if possible, but sometimes actin doesn't let it. Remember, troponin is going to be a regulatory protein. If calcium is present, and calcium will bind to troponin, it will move tropomyosin out of the way, exposing these binding sites, these black dots. And when those are exposed, myosin, which is already in this configuration state, can bind to actin and eventually pull it. So in this case, we're starting with myosin is in this configuration. Now, this does require energy to put myosin in this spot, and it requires ATP. And if you look carefully up here, you don't see ATP, but you kind of do. What is ATP? ATP is adenosine triphosphate. There it is, 3 fingers. So if ATP is going to be split, it's going to be split into ADP plus P, adenosine diphosphate 2 plus phosphate. So this is showing you, kind of picking up with the process, that we've already split ATP. We've used that energy to put myosin in its binding position, and now it's attached to actin. Super. Okay, so now what happens next? Well, notice the difference between this slide and this one, that phosphate is going to come off. And when phosphate comes off, that's going to be important in helping helping myosin change its shape. Myosin goes from this high energy shape to this kind of weak limp stage. And when it does that, it actually pulls on actin. It doesn't seem like it would pull on it, but if it changes shape and it's attached to it, it's going to pull. If I'm having a rope and I'm doing tug of war, if I change shape and pull on it, I'm not really moving my body too much, but whatever is attached to that rope is moving if I pull. In the same way, myosin, when it changes shape, since it's attached to actin, it's going to pull. And when myosin pulls on actin and pulls the sarcomere closer together, that's a contraction. So we see here that, wherever it is, that myosin has changed from up and down, if you will, to horizontal, but it's actually pulled actin as a result. And then ADP is going to be released. So let me move me up here. So what happens after that is more ATP is going to come in, and it's going to weaken that connection between myosin and actin. And then with some enzymes that are present and with this new ATP, the myosin is going to disengage from the actin. So you see down here, as new ATP attaches, the link weakens and the cross bridge comes off. So now ATP is kind of back in this limp and detached stage, but ATP is attached. That's good because ATP is going to be split. That is, we're going to split it back to ADP and P, and the energy that's released in that process is going to recock that head back into this high energy position again. Now, I have here myosin is going to bind to actin if it can, as long as calcium is present and as long as calcium is binding to troponin, and tryptomyosin is rolled out of the way, then myosin can keep binding to actin a second time, and then a third time, and a fourth time. But it's going to bind, pull, release, reconfigure, attach, bind, change shape, and pull at the same time, release. More ATP comes in. It's going to get right in shape, bind, pull, release, reattach. And it's hard to do in the video, but it's going to go through the cycle of pulling and changing shape, pulling and changing shape. And the final part here, I'm going to go ahead and put this in. The ATP that was used to position the myosin head binds to actin. So now it's going to attach. And then when phosphate comes off, it starts that power stroke. It starts to change shape to give us the full cycle. So one more time quickly here. ATP is in the right shape. We've used energy to do it. It's attached to actin. The phosphate comes off. It begins to change shape, pulls on actin. The ADP comes off. It's still going to be attached to actin until more ATP comes off, causes it to release, change shape, reattach, and then bind back to actin. So that's the contraction cycle. So why go through all that? Well, you really can't understand rigor mortis until we get to that part. So what is rigor mortis? That term literally means to stiffen at death. And while it's not exactly at death, it actually happens soon thereafter because of that same reaction, the same series of reactions we just saw. So let's see what that entails. Rigor mortis means that muscles can stiffen up, start to contract about 3 to 4 hours after death. And it's most pronounced, that is the stiffest, about 12 hours after death, and it can last up to a couple days. Rigor mortis is not where someone's going to sit up, or they're not going to blink, or they're not going to move their hands back and forth. It's where their muscles are going to gradually stiffen. So here's why it happens. The cells in the muscle fiber that are beginning to die because the person has died, calcium is going to leak out. It's going to leak out of the sarcoplasmic reticulum. There's not going to be any ATP to pump it back into the sarcoplasmic reticulum. And once myosin binds to actin, there's no ATP left to cause it to detach. So the calcium is going to cause the initial binding to happen because it's going to allow myosin to bind to actin, but with no ATP to pump calcium back into the sarcoplasmic reticulum and with no ATP to detach myosin from actin, muscles are going to stiffen up and they're going to stay that way. So since there's no ATP left to break those bonds, myosin's going to bind to actin and it just can't let go. Muscles are going to stay stiff until the actin and myosin proteins themselves begin to break down. And that's going to take a little while. I've done some forensic stuff and there's sort of this little, I don't think it's, expression, but you can look at somebody in several different ways. A body can be warm and not stiff. Okay, why would a body be warm and not stiff? Warm, it's still going to be warm if the person has recently died, and not stiff because they've not entered rigor mortis yet. So in this case, the person's probably been dead less than 2 or 3 hours. They're still warm and they're not stiff. They can be warm and stiff. That means they have not died too long ago, but they died long enough ago they've actually started rigor mortis. So this person may be having died 5, 6, 7, 8 hours ago, depending on environmental conditions and how well they're dressed and how much clothing they have on, etc. A person can be cold and stiff. That is, they've died long enough ago that they've actually lost all their heat, but they are still stiff because they're still in rigor. And then cold and not stiff. They've died, they've gone through rigor, and now the rigor is beginning to dissipate because the muscles themselves are breaking down. You get beyond about 48 hours and then you start getting into some substantial tissue breakdown and some decomposition. So if you hear on the news that someone has found a body and they estimated the person died 12 hours ago or within the last 2 days, they're probably basing that on body temperature and on rigor. There are other ways of determining when somebody died to estimate that. You've probably seen CSI, and they can look at liver temperature and potassium levels in the eye, and there's other measures you can use to estimate time of death. But rigor actually is following the physiologic process of muscle contraction, and since we understand what's going on there, we can understand and predict when someone experiences rigor about how long ago that person died. So one more time, we're going to see myosin binds to actin because myosin can, and myosin is going to stay bound to actin as long as there's no ATP available. When somebody dies, they stop making ATP. So if you have, we'll go back for a second, if you have, in this case, there it is. If you have the attachment of myosin to actin, but there's no new ATP coming in to break that bond, it stays attached. And if it stays attached, that's rigor mortis. Okay, that wraps up this video. Another one's coming. Thanks.